Hello students, I am educator Dr. Hina Devnani and today we are going to study chapter number 10 of 11th standard, that is cell cycle and cell division. This is the second part of the chapter. We have already studied the first part, which included mitosis. The first part also had details about the cell cycle, the phases of the cell cycle like interphase and the M phase. Interphase included G1 phase, S phase and G2 phase. Now today we'll be dealing with meiosis, that is another form of M phase. M phase includes mitosis as well as meiosis. Mitosis was an equational division, whereas meiosis is also called as reductional division. So let us see what is meiosis. As you can see here, it's written, meiosis is also known as reductional division. Why? Because it reduces the number of chromosomes to half the parent cell. That means the daughter cells will have half number of chromosomes as compared to parent cell. If parent cell has 46 chromosomes, the daughter cells will have only 23 chromosomes. Meiosis occurs in two phases, meiosis I and meiosis II. Meiosis occurs only in the gamete cells like sperms and eggs, that is reproductive cells, whereas mitosis occurs in all the somatic cells. Let's have a look at this figure first, then we'll be dealing with all the phases in detail. This is meiosis I and this is meiosis II. Meiosis I includes prophase I, metaphase I, anaphase I and telophase I. Now in telophase I, two cells have been formed. You can see here two cells. Now these two cells will undergo meiosis II. So we'll have prophase II, we'll have metaphase II, anaphase II, and then telophase II. In telophase II, four cells will be formed. When you have a look at this image, you can see here the number of chromosomes have become half. You can see this is single, right? Here it was double. Here there are two blue and two red cells, red chromosomes. Here there is one blue and one red chromosome. So the number has reduced. It became half the number of chromosomes as present in the parent cell. Now there is one difference here. In mitosis, the phase prophase was very simple, whereas in meiosis, this phase is very complex. Why it is very complex? Because it consists of 5 subphases. As you can see here, these are called as lactotine, zygotine, pectine, diplotine, and dikinesis. These 5 phases were not present in mitosis. There is a significance of this. What is the significance here? Meiosis occurs only in the reproductive cells, and reproductive cells means the cells which are going to the next generation. So in the next generation, you might have studied in 11th standard that variation is very important, right? So in the next generation, there should be some variations in the genes as compared to the parent cell. So the cells and the chromosome need to carry out recombination process. So to carry out recombination process, these 5 phases are very important. In somatic cells, recombination doesn't occur. So prophase I is not so complex. But in meiosis, the prophase I is very complex to carry out recombination. Here you can see recombination. The blue chromosome is having one part of pink chromosome, whereas the pink chromosome is having one part of blue chromosome. This is called as recombination. And this recombination occurs only in the reproductive cells, so that a new variety of chromosome can enter into the next generation, and the next generation will have variations, and variations always support the evolution process. So it is very, very important. You might have studied in 10th standard. These are the phases of meiosis I, prophase I, metaphase I, anaphase I, and telophase I, and prophase I consists of these 5 phases. Let us start with prophase I. It is the longest phase of meiosis I. During this phase, homologous chromosome pairs and DNA segments are exchanged, and this is known as recombination. As we have seen in the figure right now, it is further divided into the following. First, we'll start with lepto gene. This is the first stage of meiosis I. Each individual chromosome exists with 2 sister chromatids. You can see here, they have 2 sister chromatids. Elements of synaptonymal complex assemble. That is not visible in this diagram, but synaptonymal complex means some thread-like structures will be formed, which will bring these two homologous chromosomes together. Slowly and slowly, these chromosomes will start to condense. In this figure, it is not clear, but you can see the size of these chromatid is very thin. Slowly and slowly, they will start to condense, and this will become thick. Condensation and coiling will start. Here you can see these chromosomes are condensed chromosomes. This is the second phase, which is known as zygote. Chromosomes align as homologous pairs of chromosomes. These two chromosomes have come close to each other. They have aligned together. This has become a homologous pair of chromosomes. And here synapses have occurred. When they come together, that means synapses have occurred. And this synapses occur with the help of synaptonemal complex. Synaptonemal complex was formed in the first phase. The paired chromosomes are known as bivalent or tetrad. Now they are known as bivalent because two chromosomes are there, or they are also known as tetrad, having 4 chromatids. The third phase is pectitin. It is a very important phase because in this phase, crossing over occurs. What is crossing over? Here you can see X-like structure will be formed and X is formed over here. This is called as crossing over. When one arm of both the sister chromatid of both the homologous chromosomes come towards each other, they cross over each other, and the place at which they cross over each other is called as chiasmata. So here this point of junction is known as chiasmata, and this is the main point from where the exchange of segments will take place. This is the fourth phase, that is diplotin. Now, after crossing over, what will happen? The homologous chromosomes will go away from each other. They will start to separate. And when they start to separate, that means the synaptonemal complex will start to disassemble. Initially, we have seen in leptotin, synaptonemal complexes assembled. So the two chromosomes came towards each other. And now the synaptonemal complexes will disassemble. So the two chromosomes will go away from each other. But the thing that you have to remember here is the chromosomes will remain bound at the junction where the chiasmata was formed. So they will remain bound to each other from this place, whereas the rest of the chromosomes will separate. This is the fifth phase of meiosis I, prophase I, dikinesis. Here the chromosomes will condense further so that all the 4 parts of the tetrads will be visible. Here you can see it is very clearly visible. Exchange has already occurred. From here they will separate with each other. And now what will happen? All those events will occur which had occurred in prophase of mitosis, like the disappearance of nucleolus, the disappearance of nuclear membrane, and the formation of spindle fibers. All these things will begin here. And now it will behave like the same phase, like prophase of mitosis. Then we have metaphase I. Metaphase I is similar to the metaphase of mitosis. Homologous chromosomes will be aligned on the metaphasic plate or equatorial plate. The replicated chromosomes are joined together via protein, which is known as cohesin. They will be joined together. The replicated chromosomes means the chromosomes whose copies have been made. They will be joined together via a protein which is known as cohesin. Remember this name. Anaphase I. This is also similar. As the kinetochore microtubules shorten, homologous chromosomes are pulled to the opposite poles. If you remember chapter number 8, you will know what is kinetochore. Kinetochores are present on the sides of the center part of a chromosome, near the centromere. So here what will happen? The spindle fibers will attach through the kinetochores, and in anaphase, the spindle fibers and the microtubules will start to become short. And when they are shortened, when they become short, they pull the chromosomes towards the opposite poles. They split the chromosomes and the chromosomes are pulled towards the opposite poles of the cell. The cohesin protein will degrade from the chromosome arms while remaining safe surrounding the centromere. Cohesin protein will remain near the centromere so that the two arms are attached to each other, but the chromatid pairs will be away from each other because from there cohesin protein, remember cohesin protein, which is responsible for binding these arms, this cohesin protein will degrade. So the sister chromatids remain together while the homologs of the sister chromatids segregate. That means they separate. In telophase 1, what will happen? Each daughter cell will now have half the number of chromosomes as compared to the parent cell. The microtubules forming the spindle fiber will disappear. The chromosomes will form chromatin again, and sister chromatids will remain attached to each other. So this is the end of meiosis 1. Now what will happen? Meiosis 2 will occur. Meiosis 2 is the second meiotic division. It is similar to mitosis, but the genetic results will be different. Why? Because already recombination has occurred, and this results in the formation of 4 haploid cells. What is the meaning of haploid cells? Haploid cells means the cells having one set of chromosomes. Normally, in human cells, in all the somatic cells, we have 2 sets of chromosomes. That means our cells are diploid. Mitosis occurs in the diploid cells. That means 2 sets of chromosomes. We have 23 pairs of chromosomes. So we have 2 sets. In one set there are 23 chromosomes, in the other set there are 23 chromosomes. But here what will happen after meiosis? The number of chromosomes become half. So instead of 2 sets, there will be only 1 set. So when one set of chromosome is present, it is called as haploid cell. So in meiosis 2, at the end of meiosis 2, 4 haploid cells will be formed. The 4 main steps of meiosis 2 are prophase II, metaphase II, anaphase II, and telophase II. We'll start with prophase II. Nucleoli and nuclear membrane will disappear. Shortening and thickening of the chromatids will occur. Centrosomes will move to the poles and spindle fibers are formed for the second meiotic division. This is exactly similar to the prophase of mitosis. Metaphase 2, centromeres are present with the 2 kinetochores that are attached to the spindle fibers. Spindle fibers will be formed on the opposite poles of the cell, and they will attach with the kinetochores. Metaphasic plate is rotated by 90 degrees when compared to meiosis 1, perpendicular to the previous plate. This point you have to remember. Whatever has occurred in meiosis 1, in the metaphasic plate that have been formed in meiosis 1, now as compared to that plate in meiosis 2, the metaphasic plate will be perpendicular to the previous plate. So you have to remember this point. Anaphase 2. Anaphase 2 is accompanied by sister chromatid segregation. The sister chromatids will separate, they will go to the opposite poles. The remaining cohesin will be degraded so that sister chromatid segregation can occur properly. Telophase 2. This is like telophase 1. Decondensation of chromosomes will occur. That means the chromosomes will now again turn into chromatic fibers. Nuclear envelope will reorganize. The membrane will become proper. It will be arranged again. And cleavage or cell plate formation will start to produce 4 daughter cells. Till here, the karyokinesis is completed. And from here, when the cleavage starts, cytokinesis will occur. If you remember mitosis, we have done already. Karyokinesis, that means nuclear division. Once the nuclear division is over, cell division starts. So here also nuclear division will be over. And now with cleavage in animal cells, cleavage is formed, and in plant cells, cell plate formation is there. So this both will start so that 4 daughter cells are produced. And all the 4 daughter cells will have haploid set of chromosomes, whereas their parent cell had diploid set of chromosomes. So the chromosomes have become half. What is the significance of meiosis? It is important for maintaining the chromosome number generation after generation in case of sexual reproduction. Okay, now how does it maintain the number? Meiosis occurs in sperms and eggs. Sperms will have haploid chromosome number. Let's take the number, exact number, 23 chromosomes. The egg will also have 23 chromosomes. When fertilization will occur, sperm and egg will fuse together. Fusion will be there. So when fusion is there, 23 chromosomes from the sperm and 23 chromosomes from the eggs will combine together and form 46 chromosomes. So the next generation will have 46 chromosomes. The zygote will have 46 chromosomes, and hence the number of chromosomes will be maintained. Second significance of meiosis is it is important in increasing the variation in population. As I said earlier, variation is very important. It is a basis of evolution, and it is very important to survive in nature, to survive in the environment. So this variation occurs because of meiosis. These two are the important points of meiosis. So here we complete with the chapter. It's a very easy chapter. And if you want to remember all the things or all the points of all the phases of this chapter, you need to work hard on the diagrams. If you're thorough with the diagrams, you'll easily be able to write the points. So I hope you all have understood. If you have any doubt, you can contact me. Until that, take care and have a nice day.